Okay, good morning.
Good morning.
I open this academic session in which Akhil Konkoth will defend the academic thesis Multifaceted Role of Microvesicles in Thrombo-inflammation, Role of Microvesicles in Haemostatic Imbalance in Sepsis. May I invite you to present a summary of your studies and the conclusions of your thesis.
Thank you. Is my slide visible? I hope my slide is visible.
Yes, it's visible.
Yeah. So good morning, everybody. Dear Pro-Rector, dear members of the Corona and friends, thanks for being here. My name is Akhil Antony. I would like to give an overview of my PhD thesis, which is entitled Multifaceted Role of Microvesicles in Thrombo-inflammation, Role of Microvesicles in Haemostatic Imbalance in Sepsis. So extracellular vesicles are membrane-bound vesicles. They are primarily classified into three main types based on the biogenesis. The first type is exosomes, which has a size ranging from 30 to 150 nanometer. They are generated within the multivesicular bodies, and when the multivesicular bodies fuse with the cell membrane, it releases the exosomes into extracellular space. The second type is microvesicles, which has a size ranging from 100 to 1000 nanometer, and then they are formed as a result of a budding of plasma membrane. The third type is apoptotic bodies, which has a size ranging from 500 to 5000 nanometer. They are formed when the cells undergoes the process of apoptosis. Next, I will briefly explain the role of EVs in hemostasis. EVs have long been regarded as the procoagulant entities, mainly because they express anionic phospholipids like a phosphatidylserine, and they also express tissue factor, mediating the assembly of coagulation factors and leading to activation of the coagulation cascade. The EV-dependent tissue factor activity can be measured. For example, when the whole blood was stimulated with LPS, it significantly increased EV-dependent tissue factor activity. And the tissue factor positive EV is derived from cells, such as the BXP3 cancer cell lines. It can be captured from biological fluids, and it can be labeled using ExoView technology. The procoagulant activity of EVs have been explored in several in vivo thrombosis models. For example, in a carotid artery laser-induced injury model, when infused with monocyte EVs with high tissue factor activity accumulated at the site of a thrombus formation. So in this image of a growing thrombus, the red color represents the platelet, the green color represents the tissue factor, and the yellow color represents both platelets and the tissue factor. In another study which used a deep vein thrombosis mice model, the tissue factor positive EV is derived from L3.6PL cancer cell lines. When it is injected, it positively impacted thrombus growth. And when these EVs were treated with the anti-tissue factor antibody, it significantly reduced the thrombus weight. So far, we have seen the procoagulant activity of EVs. However, especially from our lab, we have demonstrated that some subset of EVs lead to dissolution of a clot that is mediated through urokinase plasminogen activator and its receptor, as well as the tissue plasminogen activator. So this distinct opposite effect of EVs when it comes to hemostasis seems to be a balance between both procoagulant and profibrinolytic activity, which allowed us to introduce the term EV coagulolytic balance, or simply EVCLB. And in my thesis, I investigated how this EVCLB is modulated in the context of a sepsis pathology, as well as in the reapheresis treatment procedure in patients with the peripheral arterial disease. So my thesis had two general objectives. The first one was to perform a comprehensive evaluation of the procoagulant and or profibrinolytic properties of a different subset of EVs purified from human cells or directly isolated from plasma of healthy individuals and sepsis patients. The second objective was to evaluate the generation of EVs with high fibrinolytic potential by neutrophils induced by biopolymer membrane filters during reapheresis session. Due to time constraints, I will focus on the first objective. That's the major part of my thesis. So sepsis and septic shock has a high epidemiological prevalence. It accounts for almost 1/3 of all ICU cases, leading to 11 million deaths every year. Its pathophysiology begins with an exacerbated systemic response to infection, which is characterized by activation of endothelial cells and leukocytes, which then leads to dysfunctional coagulation. That means there is an overactivation of coagulation pathways and dysfunctional fibrinolytic pathways, which then culminates into disseminated intravascular coagulation. So the procoagulant EVs can exert a deleterious effect in sepsis patients. For example, one study demonstrated there was a significant increase in EV-dependent thrombin generation in patients who died as compared to the patients who survived. However, very recently from our lab, we demonstrated that granulocyte EVs with the high plasmin generation capacity had a protective effect in septic conditions. And when these EVs were injected into septic mice, it removed microthrombi from vital organs, and that was associated with the significant survival of the mice. So with that said, the hypothesis of my project was the cellular origin and the pathological context driving the biogenesis of EVs could determine the distinct functional profiles of EV-CLB. Next, I will briefly explain the method, cell preparation, and EV purification. First, the human neutrophils and the monocytes were isolated from whole blood using magnetic activated cell sorter. And the neutrophils were stimulated with the TNF-alpha, and the monocytes were stimulated with the LPS overnight. And the EV containing culture supernatant was subjected to a series of centrifugation to remove cell debris and apoptotic bodies. And EVs were concentrated using ultrafiltration. And then they were subjected to size exclusion chromatography to remove any soluble contaminants like a protein. And finally, EVs were isolated, pelleted using ultracentrifugation. Next, I used a human cardiac microvascular endothelial cells. They were stimulated with TNF-alpha for 24 hours, and the EVs were isolated using the same method as this. And finally, the platelets were isolated from the whole blood, and they were stimulated with thrombin to generate EVs. EVs were pelleted using ultracentrifugation. Next, I performed a set of characterization of these EVs, different subset of EVs based on a minimal study of information on extracellular vesicles. First, the TM images showed the presence of a lipid bilayer enclosed all the subset of EVs. And next, the flow cytometry revealed the small events that is a representative of a different subset of EVs. And next, the Western blot analysis revealed all the subset of EVs expressed alpha 5 subunit of integrin. That is indicative of a cellular origin of these EVs. And the Western blots also revealed the purity of this different subset of EVs, that they were devoid of any contamination from soluble proteins like albumin, except for PDVs, there was a negligible contamination from albumin. And finally, the Q-nano analysis revealed the homogeneous distribution of size distribution of this different subset of EVs, with the mean diameter was between 150 to 177 nanometer. Next, I performed a series of functional assays to evaluate the coagulability balance of EVs. The procoagulant activity was evaluated using factor generation assay, thrombin generation assay, and clot formation assay. And the profibrinolytic activity was evaluated using plasmin generation assay. So the MDVs and EDVs from unstimulated cells, they supported low but detectable level of factor generation, and that was significantly increased in inflammatory conditions. And interestingly, the factor generation by these EVs were inhibited using an anti-tissue factor antibody, as well as in the absence of a factor 7, which indicates that a tissue factor pathway is involved in the factor generation by these EVs. And furthermore, the factor generation by these EVs were inhibited using annexin 5, which indicates that the phosphatidylserine is also involved in factor generation. But on the other hand, treating these EVs with an anti-tissue factor pathway inhibitor antibody did not influence their factor generation. In contrast to MDVs and EDVs, the NDVs and PDVs did not support a factor generation. And again, the procoagulant activity of these EVs were further confirmed using thrombin generation by spiking these EVs into EV-depleted plasma. So the levels of thrombin generated by MDVs and EDVs were increased under stimulated conditions. And for the MDV generated thrombin was significantly inhibited by using an anti-tissue factor antibody, but on the other hand, it was only partially inhibited for EDVs. And in contrast to MDVs and EDVs, and both NDVs and PDVs did not have any thrombin generation capacity. Next, I looked into the ability of this different subset of EVs to influence the fibrin clot formation by spiking these EVs into EV depleted plasma. So the SEM images revealed the ultrastructural differences in the clot formed by these different subset of EVs. And in this image, you can see that the clot formed by MDVs and EDVs from stimulated cells, they formed a very compact and dense clot. And that was associated with the significantly decreased diameter of the fibrin fiber and the porosity, that's the space between the fibrin fiber. So far I explained the procoagulant activity of EVs, and next I looked into the other side of the EV-CLB, that is the profibrinolytic activity, and which for that I used a plasmin generation assay. And here the NDVs, MDVs, and EDVs, both from stimulated and unstimulated cells, they cleaved the plasmin, generated plasmin in a concentration-dependent manner. And interestingly, the plasmin generated by NDVs from unstimulated cells were slightly higher than the other types of EVs. But in contrast to the PDVs, it did not have any plasmin generation capacity. So to conclude the CLB profile of EVs in vitro, both MDVs and EDVs more like had a CLB profile of profibrinolytic under unstimulated conditions. But this profile was significantly shifted to like more procoagulant under inflammatory conditions. But on the other hand, NDVs had more like a profibrinolytic CLB profile under both stimulated and unstimulated condition. And lastly, the PDVs did not have any procoagulant or profibrinolytic capacity. Next, to evaluate the EV-CLB profile in vivo, I immunocaptured different subset of EVs circulating in plasma that is collected from healthy donors as well as the septic patients. Then I performed the factor generation and plasma generation assay on these EVs. So in the healthy plasma, only MDVs had low but a significant level of tissue factor activity or factor generation, and that was increased in septic plasma. And interestingly, EDVs, NDVs, and PDVs had a factor generation in some septic patients. Next, I evaluated the plasma generation capacity of these EVs. And in healthy plasma, only NDVs had plasma generation capacity. And when it comes to septic plasma, the result was the plasma generation by NDVs were more heterogeneous. And finally, I calculated the EV-CLB. That's the ratio between them both EV procoagulant and the profibrinolytic activity, which is an arbitrary unit. And only MDV EV-CLB was a more procoagulant actually, and that was a significantly increased in septic conditions or inflammatory conditions. And in contrast, only NDVs had a low EV-CLB, that is a representative of a very high fibrinolytic activity. And interestingly, only NDV's EV-CLB was positively correlated with the fibrin monomer, and that was negatively correlated with the fibrinogen and the platelets. So that's markers, important markers in the coagulability of sepsis. So to conclude my work, this work provides the first demonstration that the cellular origin and activation status of the cells drive EV-CLB profile. MDVs had the most procoagulant CLB with a significant procoagulant activity, and NDVs displayed the most profibrinolytic CLB profile with high plasmin generation activity and significantly correlated with the coagulopathy markers. And finally, the EV-CLB may represent a potential biomarker reflecting the hemostatic imbalance in a sepsis. So I would like to thank my team in the Marseille and the Maastricht, our lab head in the Marseille, Professor François Digna-Georges, and my supervisor from Marseille, Professor Romeric Lacroix, and my supervisor from Maastricht, Professor Rory Conan, Professor Tillman Hacking, and my supervisor from Biostatics, Dr. Philippe Poncelet. And I would like to thank all other team members from Maastricht and Marseille. So thank you.
Thank you for your presentation. It's now time to start the opposition. And the opposition will be opened by the chair of the assessment committee, Professor Dijkgraaf, Professor of Biomimetic Chemistry at Maastricht University. Professor Dijkgraaf.
Yeah. Thank you, Pro-Rector. Well, dear candidate, I first would like to congratulate you and your promotion team on the completion of your PhD thesis To me, it was really a joy to read it, and I learned a lot about the microvesicles and also the ways they can be formed. So yeah, thanks for that. But of course, I do have some questions, and I first would like to start with you and go to page 15. There you state that various microvesicle releasing inhibiting substances like calpeptin, imipramide, etc., could be used as a therapeutic strategy. Well, this is actually in contradiction with what you write on page 12, because there it's written that that you want to deliver some cargo, so some bioactive molecules to recipient cells. So what is now the best therapeutic strategy in your opinion? Is it inhibiting of the release of microvesicles? And I'm stating microvesicles, but actually I mean also the exosomes, all the extracellular vesicles. Is it the inhibition or the promotion of the microvesicles?
I think it's possible to look into both the aspect of these microvesicles or EVs in general, where we can actually modulate their release or upregulate the release of to kind of modulate certain cellular functions, including like a thrombus, their functions on a thrombosis or inflammation. So like I have mentioned in my thesis as whether it is by pharmacological means or like gene regulation, like a knockout of a certain genes, as it can modulate the release of a certain types of EVs, so which can actually regulate when it comes to disease types. But it's also important as especially from our lab, we are actually working on this thrombo-inflammation or like modulating thrombosis. So we are mostly interested in injecting certain types of EVs, especially the fibrinolytic EVs, because we are very, very much interested in neutrophil EVs or NDVs, because of its high fibrinolytic potential. And from our lab, we have demonstrated that EVs is generated from HAP1 cell lines with high fibrinolytic activity. When injected into mice with the thrombosis, it can enhance the plasmin generation or it can aid thrombolysis. So yeah, I will look into both of these aspects.
Yeah, but if I hear you well, you are more a fan of promoting extracellular vesicle release, so that you promote the formation of this. Yes.
Yeah.
But how do you foresee a therapeutic regimen in patients? Because usually you have to, if you want to have a good therapeutic compound or other thing, you need to have a specific targeting. How are you planning this? How are you what is your strategy to tackle this problem?
In fact, we can in the lab actually we can produce this large quantity of let's say talk about these NDVs with high fibrinolytic activity. We already have started work on this aspect actually back in Marseille in collaboration with the other labs actually. So we want to look into this fibrinolytic activity of these NDVs as a model of using septic patients. So in the initial aspect, we use septic mice and injecting these EVs into septic mice, whether these EVs can actually remove microthrombi. Especially we have already demonstrated that in septic mice, as it can remove microthrombi from vital organs. But we want to extend this aspect into septic induced thrombo-inflammation in the brain or ischemia in the brain, whether injecting these particles into the vasculature, whether it can adhere to this thrombi, and in reality, it can actually initiate the thrombolysis.
Yeah. I do agree with you that if you really have a look at sepsis or the removal of thrombi, this works. But do you also investigate off-target effects? Because I can imagine that you really are not only targeting.
Yeah, it could be as but there are other possibilities as whether we can actually deliver these vesicles in target specific way. So we will have to modulate or bioengineer these vesicles so that by modifying its surface molecules, so that it can be delivered so that it will only go to the target we want it to go.
And are you not afraid that because you also write somewhere in chapter 1 that the microvesicles carry various bioactive molecules? Are you not afraid that these other bioactive molecules can cause other diseases or inhibit
Could be
other?
as like that's why we need to choose as like the vesicles that coming from a certain types of cells. So I talk about these neutrophils, and I think neutrophils are more fibrinolytic as well as this have like a more anti-inflammatory properties actually, like when stimulated with a certain kind of agonist, so that it does not have this pro-inflammatory effect. So I assume actually like it's very safe to inject these vesicles are for this is certain types of. I'm not sure whether it can be like injected for other types of disease, but I think in a thrombo-inflammatory conditions or a thrombus formation, I think it is very, but it needs to be tested anyway. Like
I would definitely recommend you this to test these, because I think that the presence of other biomolecules can be a big burden to, especially to patients that are having sepsis. This is a very vulnerable group.
And I also analyzed some like these pro-inflammatory markers in a different subset of EVs, and what I found was actually in the neutrophils we do not have that much of a pro-inflammatory markers as compared to the MDVs, the monocyte EVs, and the endothelial EVs, which had like a more high pro-inflammatory cytokines when stimulated from stimulated cells. So I assume as like it's safe to inject these neutrophil EVs.
Okay, well, I'm happy with your answers, and I think I have, for the sake of time, I give the word back to the pro-director. Thank you.
Okay, thank you very much. I had some trouble with this unmuting. The opposition will be continued by Professor Morange, Professor of Hematology at Aix-Marseille University in France. Professor Morange.
Thank you very much. Dear Akhil, first I would like to congratulate you on your thesis, because you have done a great job, and particularly your presentation was very clear and interesting. I have several questions, mainly focused on the first manuscript regarding sepsis. My first point is regarding the 10 patients you have selected for with sepsis. You said in the material and methods that it comes from a very large cohort. I'd like to know how did you select those specific 10 patients?
We had this large cohort, but I selected it at random as like I did not look into the specific aspect of these patients diagnosed or like a criteria, the clinical criteria they presented. It was just a random selection of these 10 patients.
Okay.
Yeah.
And why did you use 10?
There is no specific. I wanted to use more, in fact, as like, but this isolation using immunomagnetic separation is a very laborious process, especially considering that I have to isolate these 4 different types of EVs from a plasma. It's like so. I wanted to actually like include a more number of patients as like so that it will give more data that is representative of a general representative more number of patients actually, but unfortunately I could not include more number of patients, but I just concluded with the 10 number of patients.
Okay. And I have the same question for the so-called healthy controls, the 8 subjects. How did you choose them, and where did they come from?
It's mostly coming from Marseille itself, actually, like from our lab members and the members working in the hospital. So I asked members actually like so age matched control, healthy controls that matches with the septic patients. Okay, that's mostly coming from Marseille. Yeah.
Okay, and as far as I know, it's pre-analytical conditions are very important in the setting of EVs. For each patient, you have run the one healthy control at the same time, even with pre-analytic. How did you?
Not at the same time, actually. Like so, I ran the septic patients, means like intermittently, like. Not like I ran all the septic patients at once and then the healthy controls, but it was like more mixed up.
Okay. Okay. I've got a second methodological question.
Yeah.
Why did you choose HMEC as endothelial cells? And did you try other endothelial cells to verify whether you have the same results?
Yeah, I tried because I wanted to stick on to this cardiovascular area. And I wanted to focus more on this microthrombi, especially like when it comes to sepsis or other thrombo-inflammation. This microthrombi is more important because it can lead to this shutdown of multiple organs. So I wanted to particularly choose microvascular endothelial cells. And regarding other types of endothelial cells, for this work, no, I haven't tried. Let's say like a HUVEC or other types of endothelial cells, I haven't tried. But I have tried endothelial cells from IOTA.
Okay.
And they had like a similar profile, actually, like similar to HCMEC, I would say. But this is the reason actually like I wanted to stick to microvascular endothelial cells.
Okay, thanks. One more question.
Yeah.
You say that, and you give convincing results that EVs from neutrophils doesn't play a role as, let's say, procoagulant EVs, which is, as you said, controversial because some say that it's very important. And we all know that neutrophils play a key role in thrombosis. I'm surprised by the thing that you said that 50% of the 10 patients, in fact, present tissue factor, at least a procoagulant dependent tissue factor activity. Do you know why are they different from the 50 other percent? Did you check for some specific reason for having those procoagulant activity?
I haven't specifically looked into that matter, but it's already known that as like neutrophil EVs can form aggregates with other types of, let's say like a procoagulant EVs from a monocyte origin or endothelial origin. And it's also known that as like a tissue factor can be transferred from other cell types, like procoagulant cells to neutrophils or neutrophil EVs. That might explain as like to some extent why neutrophil EVs have this procoagulant activity. But some studies have shown that as like neutrophils express tissue factor, but in terms of their functional activity, I presume as like nobody has shown that as like it has as a functional activity, although some studies have shown expression of a tissue factor. But I know that as like neutrophils have other roles in the thrombosis, thrombo-inflammation, that they can form this net formation, neutrophil extracellular trap. It's very much involved in the thrombo-inflammation or thrombus formation.
Okay. And going back to the 5 patients with the tissue factor dependent activity, what is your opinion?
So for the neutrophils?
Yeah, sorry. Yeah.
I assume as like it could be maybe aggregates, some aggregate formation with the monocyte or endothelial EVs. And there could be other factors as like that may modulate as like a procoagulant activity on these neutrophil EVs.
Okay, thank you. I'm done. Thank you very much once again.
Thank you, Professor Morange. The opposition will be continued by Dr. Heinzmann, postdoctoral researchers at the University Hospital at RWTH Aachen in Germany.
Thank you, Prorector. Dear candidate, also from my side, your congratulations for your thesis and your standing here. Also to your promotional team. And I'm also going to continue for your first manuscript. So it was really interesting. You also wrote a very nice book with a very nice overview of everything. But I came across your method section, and there I saw several methods that you used for EV isolation. And I found a new method that I have not used myself, and that's the magnetic method for isolating EVs. And I wanted to know your opinion about would you recommend it to someone like me who wants to maybe use it for her own research, or and what the benefits and then the drawbacks are of this type of method?
Yeah, I would definitely recommend actually, but this method is a relatively new method as compared to other conventional methods which we can use for EV isolation. One of the good important aspect, or like a very good aspect of using immunomagnetic or IMS strategy for EV isolation is that it's very fast as compared to other conventional methods. Like you can do like isolate different subset of EVs in a matter of like 1 or 2 hours from a very tiny volume of samples, whether it be like a plasma or other biological fluids. As low as like 100 microliter. I worked with 100 microliter for plasma. So that's a very tiny volume as like you can use. But the methods needs a lot of optimization. And we are not sure as like, because in a plasma or a biological fluid, so there can be different types of EVs. So you are not sure as like you are isolating a specific type, whether it be like a small EVs, exosomes, or microvesicles in that say. So it capture or pull up everything with the specific marker you use for us like to isolate EVs. Yeah, that's the one caveat I can think of.
So if you would be more prone into looking in platelet EVs inside the circulation, you would not recommend this.
Yeah. If you want to like analyze something immediately, very quick, actually, like I think it's a very good go for like.
Okay, interesting. That was for me the more technical part. For the while, I was very interested because you looked at the heterogeneous population and your coagulation balance. I was wondering, you looked into the sepsis patients. You selected 10. Did you look at also the severity and the survival rate of those patients?
Yeah, I think all of our patients in that 10 patients, that's like they survived. So some patients had like, I don't remember how many, I think maybe like 3 or 4 were like very severe. And a couple of these patients had like a high fibrinolytic activity. I cannot really say it's because of high fibrinolytic activity the patients survive. But yeah, 3 or 4 patients were in severe conditions. I think all of them like survived. Yeah.
So you can kind of like give your opinion about that you could tackle with kind of like biomarkers to see how severe those patients are and how those
absolutely. That's the main goal of our studies. In fact, although like it's only like a 10 patients I could include in my study, but from our lab, actually, like in a parallel study, and a study that was done before me, like we included this large cohort of patients, around like 225 patients. So we looked into this EV-CLB, the coagulability balance, with neutrophil EVs, with other subpopulation, like a cumulative effect, procoagulant effect of other EVs. And when we calculated the EV-CLB, actually, like it provided us a better, like a marker that we can actually like differentiate the patients based on this EV-CLB score. So when the patients had like a low EV-CLB, that's a representative of high fibrinolytic activity, these patients had a better survival rate. So that study has to yet to come out. It will soon.
Oh, good. Yeah. So what would you then advise a doctor knowing this? Would you recommend like implementing this type of isolation and to track the patient, or would you say it needs a bit more work, or what would your advice be to use this in the clinic?
Absolutely. Given the current diagnostic or prognostic as like difficulties associated with like stratifying these patients, like not only to diagnose but to come to this prognostic value, as like I think we can actually like definitely incorporate these kind of methods into like stratifying patients, and also to evaluate the treatment response on this patient. It could be definitely used. But along with other, like not this alone.
Yeah. I don't know if you look at it, but you have like this heterogenic population in the sepsis patient. Did you look at which population was more present than the other? So it would have been more platelets, would have been more endothelial, or more neutrophil populations, EVs present in that heterogeneous pool, or you did not look at that?
It's always platelet. I'm not sure as like if I think I have included that data as like the platelets. The presence of platelet EVs were significantly more as like in the patients, but still like their procoagulant activity was nowhere comparable like to the most procoagulant EVs, the monocyte EVs. Yeah, they had like a sheer number of platelet EVs in the circulation.
Would
And
you
the
then,
least was endothelial EVs.
would you then target those population as a kind of like a treatment? So to bring down those EVs, or would you say
could be possible as like we can perhaps try to like manipulate like MDVs, like monocyte EVs, which are most procoagulant. So we can perhaps like try to like reduce their levels in circulation, which could aid in thrombo-inflammation.
Okay, I'm satisfied with your answers, and I will give the word back to the prorector.
Thank you.
Thank you very much, Dr. Heinzmann. The next opponent is Dr. von Hundershausen, Principal Investigator Platelets and Clinical Studies at the Institute for Cardiovascular Prevention, Ludwig-Maximilians University in Munich, Germany. Dr. Hundershausen.
Many thanks, Prorector, dear candidate. Also, congratulations from my side. I think you did a very clear structure in your presentation as well in your thesis, and I also like the cover. So in the center, there's this microvesicle, and you have on the right and left side more or less the dark and white side, or the pro-fibrinolytic and pro-coagulant activities. And there, I think if you show this microvesicle, that there I have the first question, because your neutrophil-derived microvesicles, they show enhanced plasmin activity.
Yeah.
And this happens in several scenarios. Once you do this rheopheresis, and once it happens also in sepsis patients. So what is the mechanism that the neutrophil-derived microvesicles generate more plasmin?
It's attributed to like this receptors actually like for plasmin generation. We have this urokinase plasminogen activator, but it's neutrophils express its receptor, and neutrophils itself actually do not express actually the urokinase plasminogen activator. It has to come from other sources. So neutrophils has this PLA-UR gene, which gene is actually like translated into this urokinase plasminogen activator receptor. So it expresses a receptor, but it captures actually like a urokinase plasminogen from the circulation. And depending on the stimulus you give as like it can respond to like a various stimuli, but it's only like a certain stimuli it can upregulate this fibrinolytic activity or the expression of a UPR receptor on the surface.
So is the urokinase receptor upregulated?
So
you measure more receptor on the neutrophils and the microvesicles?
That could be one of the reasons. And in rheopheresis, I assume as like it could be like a sheer increase in the neutrophil EVs. I haven't looked into how much or how many like receptors are expressed on EVs, but we see as like a huge quantity of these EVs released into circulation after the rheopheresis session. So it could be either the number of EVs or could be both as like an increased expression of UPR on the surface.
Yeah. If you cut down the membrane and put it into the tube, and then you add the neutrophils, you see barely no effect. I think it was at least not significant. What is it then? Why do you get more plasmin activity on the microvesicles if they come from the patient after rheopheresis instead of your tube in vitro system?
In vitro, in fact, I tried actually. There was when I treated these neutrophils in the presence of this membrane, I cut open this membrane into small pieces of like 2 millimeter size, 2 millimeter length. And when I treated these neutrophils, isolated neutrophils with the membrane, it activated neutrophils and also increased the number of vesicles. So it's a kind of recapitulate what we observed in vivo in patients actually when during the treatment session, it significantly increased neutrophil EV. So I assume as like the reason, one of the main reason as like there is a neutrophil activation in the presence of the membrane, and also it increased the EVs with the fibrinolytic activity in the circulation.
Yeah, so, but we don't know, so it depends on the membrane, but we don't know what kind of...
It depends on the membrane, because these extracorporeal treatments are like, let's say like a rheophoresis or dialysis or plasmapheresis. Different hospitals use different kinds of membrane, and these different membranes are made up from different chemical substances. The one I used in the rheophoresis is actually, it's made of a polysulfone. It's a polymer, which has relatively good biocompatibility compared to like other like membranes that is made of like a ethylene glycol and or other substance, cellulose kind of membrane. And that's a previous generation of membranes actually, and they had like very low biocompatibility. And it's known that these membranes can actually like induce activation on platelets or neutrophils. Polysulfone is more like, it has a very good biocompatibility, but still induce the activation of cells.
Okay. You measured neutrophil activation. What markers are there that you can use to determine neutrophil activation, and what is their functional role?
I used CD11, 11B. It's an integrin, actually, MAC1 integrin, and it's very, it plays an important or a crucial role in like adherence. It migrates to like a different inflammatory site, thrombocyte. It helps neutrophil or even present on EVs. So it helps the EVs or cells to adhere to thrombus formation, and it can initiate its thrombolytic. And I think at least I have read a couple of studies as like this integrin also work in synergy with the UPR. It's a receptor to initiate the thrombolysis. So I think it's important not only for adherence, but I think it's important for thrombolysis as well.
And are there other activation markers that you know of?
I can use CD66B, but it's not an activation marker, but I think it can be increased in activated neutrophils.
So for instance, there's L-selectin, that would be one. Do you know what happens if neutrophils get...
This one actually, PSGL, could be used, or yeah.
You know what happens if you activate the neutrophil? Does it go up or does it go down?
Which one? Which marker?
The L-selectin. If you...
I think it can increase, but I personally haven't looked into that.
Yeah, there's shedding, so there is more or less, although it's as well an adhesion molecule, it may get upregulated, probably yes, but in the end, it gets cleaved off the cell surface and you measure it, and in the end, the expression goes down. And then as well some release factors, so the neutrophils release as well substances that you could measure. Any idea what is a super known molecule derived from neutrophils? Soluble mediator?
Could be. It's in some scenarios, as some cases, depending on what kind of a stimulus you give, it can upregulate some pro-inflammatory molecules. I myself did this Luminex assay, and when I looked into these different types of vesicles, as like neutrophil EVs that is derived from cells stimulated with the TNF alpha, it had this upregulation of IL-8. IL-8 is a very important chemokine. Actually, it's important for like a chemoattractant of neutrophils and other lymphocytes towards the thromboinflammatory side.
I don't know how much time do I have. Do I have more time for another question?
I think your time is
over.
About up.
Okay, then thank you very much. And then I hand over to the next reviewer.
Thank you very much, Dr. van Hondershausen. The next opponent is Dr. Roest, Clinical Director of the Synapse Research Institute in Maastricht.
Dear candidate, it was a pleasure to read your thesis. And for me, as non-specialist on microvesicles, it's also an opportunity to learn new things. As you already indicated, your main work in this manuscript is the article 1 on page 71. So I'm not the only one, but I like to go into this manuscript. Well, the first thing I did in this manuscript was to go through the figures to see if I could understand it, if I could follow the figures, and if I could draw conclusions from them. And those figures really made me enthusiastic about the research, because I thought, yeah, these are really clear results. You can straightforward, and I can draw my conclusions from those figures. But after that, I started reading your abstract and your conclusions, and I thought, oh well, I thought your results are very clear, and the conclusions in your abstract and in your discussion are very vague. So I like to go with you through the different figures and see what kind of conclusions I have drawn and what kind of conclusions you draw, and then see if you agree with me that you can be a lot more straightforward in the conclusions of your manuscript. Okay. And after that, I like to make a short step to the clinical studies, because I think it's very important to be very clear in your conclusions. So let's go to page 83. And as I said, I think that was for me that figure was very clear. But what is your conclusion about this figure?
I'm not sure as like what figure, because I don't have a thesis with me. Like if you could just
that's the figure in which you did the factor 10 assay, the tissue factor dependent factor 10 assay with the different types of extracellular vesicles.
Vesicles, yeah. So like when I presented the thesis, actually, like so from that image, from that figure, you can infer that the monocyte and endothelial EVs, when stimulated, actually it have this significant factor generation. But on the other hand, I did not find as like for a neutrophil and platelet EVs have any factor generation activity, at least in the in vitro, like an assay, a buffer-based assay. So that's what I infer as like depending on the stimulus you give, monocyte and endothelial cells can produce as like more procoagulant EVs.
Yeah, that was also exactly my conclusion.
Yeah.
If I continue on that, does that mean that platelet microvesicles and are not procoagulant?
Not really as like, but in this buffer system as like we really tapped into this specifically to tissue factor. I think from the literature, it's almost clear as platelets to some extent they do not express at least a functional tissue factor, but they are very procoagulant as like one of the reasons is because of its a sheer number in circulation, and it's also procoagulant because of the exposure phosphatidylserine. That's one of the important molecules like which can exert or like to play a role in procoagulant activity for these platelets. I haven't personally looked into.
But still, it's an important finding that you confirm, because I think there's still debate whether there is tissue factor in platelets or not.
Yeah.
You clearly show here there's no tissue factor on the platelet extracellular.
But some studies have demonstrated expression of a tissue factor, but I think these studies have not looked into functional activity. They haven't done any functional studies on. They have done like they have shown mRNA expression or tissue factor expression on the surface, but it does not translate into functional, because this tissue factor can be like in a cryptic, like inactive form as like. So I think the studies have to mandatorily have to look into functional studies to know if it has a like active tissue factor on its surface.
So you think that if you rewrite your conclusion, that you can say the NDEFs and the NDEFs, so the neutrophils and the monocytes, the extracellular vesicles, they express functional tissue factor, and the EDEFs, I'm sorry, and the neutrophil DEFs and the platelet DEFs, they don't express functional tissue factor.
Yeah, could be that. I think I have put that into discussion perspective. I think I'm not sure. I think I have discussed that in the discussion session. That it could acquire. It's also like it could acquire tissue factor from other cellular sources. That could also be like a possible.
Okay. well, then in the next figure, you confirm that with thrombin generation, which is very elegant, I think.
Yeah.
Then I move to the figure 6, and that's the figure in which you measure the plasmin generation.
Yeah.
And can you summarize those, your findings?
That's I only find that this among these different subset of EVs, only EVs from endothelial, sorry, neutrophil, monocyte, and endothelial cells had as a plasmin generation capacity. And it's known that from my experiments and the experiments from others from our lab that neutrophils express urokinase plasminogen activator receptor. Monocyte kind of express its receptor as well as a urokinase plasminogen activator. And endothelial cells or EVs, they express the receptor as well as the activator, and can also like express tissue factor, tissue plasminogen activator. So it's obvious that actually like given that they have this urokinase receptor and its activator, they can actually like produce plasmin. They can cleave plasminogen to plasmin. But for platelets, we and others haven't demonstrated expression of either UPR receptor or a UPA. But platelets, I think they can act as a substrate. The plasminogen, I think, can bind to platelet membrane, and maybe the vesicles or cells from other cells, let's say like a neutrophil or a monocyte, can cleave plasminogen that is bound to like a platelet surface.
Yeah, but you now show that platelets derived extracellular vesicles, they don't have the capacity to form plasmin.
After like a extensive purification, I don't think as like they have any plasmin generation.
So if they do it, then it's pollution or interaction with other cells.
Yeah.
For me, that's a very important conclusion, because if you ask me, then I would always say, well, platelets may be involved, but I don't know. And when I saw these figures, I thought, well, that's a nice conclusion. And if I read then your conclusions, I do not find it back.
Okay,
and I think it's very important, because if you then go to your next step, your
and you keep it very short, Dr. Roest? Short question.
Okay, because in your sepsis patients, you see that the MDEFs are the only extracellular vesicles that are distinct from the controls. So if you
can you please repeat that?
In the sepsis patients, you see that the MDEFs are the only increased extracellular vesicles, and you know those are the procoagulant ones. And the neutrophils are not. So if you have a clear mechanism for each derived particle, then it's easier to draw conclusions in your clinical studies.
Yeah, I would assume as like, at least from my study and studies from others, as like the monocyte or monocyte EVs are most procoagulant. And not only that, that depending on the stimulus, as like whether it be like LPS or other pro-inflammatory stimulus, as like it can significantly upregulate the expression or activation of this tissue factor into active form, or it can actually express more tissue factor on its surface. So, and also can increase the number of EVs released into circulation. So there are different aspects which can significantly promote the procoagulant activity for these vesicles. So that's one. And on the other side, I assume as like the neutrophil EVs are more pro-fibrinolytic.
Okay, so I think we agree on that. The discussion can be more clear, and for the rest, I like to give the word to the prorector.
Thank you, Dr. Roest. We come to the last opponent. That's Dr. Nagy, assistant professor at the Department of Biochemistry, Maastricht University. Dr. Nagy.
Thank you. First of all, dear candidate, I would like to also congratulate you and your supervisory team on this nice achievement. I really enjoyed reading it. And as it was mentioned before, I mean, for me, that was also a good learning opportunity as well, because before I a bit neglected and EVs because it was too complicated, but that's a nice overview. And I also have some questions regarding your chapter 1. Where, given my background, I was actually really excited to read about thrombin generation factor 10 assay. And so you on the page, you don't have your thesis, so I try to
Okay, yeah,
question really clear.
yeah.
So first of all, page 83 on the bottom, you mentioned lag time in nanomolar. I think that's just a typo, but maybe for later you need to correct that.
Okay, yeah.
But my actual question is regarding your thrombin generation, when you performed it with the various EVs, and you could show that if you added tissue factor inhibitor or antibody, then you actually got abolished thrombin generation.
Yeah.
And then when I see at the thrombin peak, that's obvious that there is no thrombin. But when I took a look at your lag time, you indicated in both the stimulated and unstimulated tissue factor antibody treated samples, lag time of 0, which would mean that you have already thrombin generation when the measurement starts. So how the curve look like in this case?
Curve was like its peak was significantly reduced, and it shifted more towards like a right. So that means that's the indication of like its lag time is actually more.
Yeah, but did you have actually completed curve for them, or you just had a flat line?
Not a flat actually for the EVs from a stimulated condition. Definitely not flat, but it was almost towards the bottom.
Yeah, because that's in the lag time, what you indicate on the panel B with the actual graphs, you indicate lag time of 0. So
yeah, it almost completely like completely nullified.
But then you actually don't have lag time, so you can't say lag time of 0 because that would mean that you already have thrombin generated at the time.
That makes sense. Yeah.
So another question regarding factor 10A, you already discussed before with Dr. Roos. But my specific question is, you also with the sepsis patient, when you measure them, you also calculated tissue factor independent activity based on your factor 10A activity data. But it made me a bit wonder because you use also for this a buffer-based system. So you added back purified factor 10, factor 7A in certain conditions and your EVs. So where is this residual activity really coming from? Because your substrate is supposed to be really specific for your factor 10A. So if you inhibit, if you don't have tissue factor, then what else then activates?
It could be coming from like other procoagulant molecules like phosphatidylserine, of course, definitely play a role in.
But then do you propose then the phosphatidylserine activates directly factor 10, or how does it?
It could be like a scaffold for like other factors can bind, but different types of EVs like they have a contact dependent pathway activation, like they can use a factor 12 and 11 or factor 9 in that case, like especially for platelets and platelets definitely it can use the contact dependent pathway to generate thrombin or a factor generation.
Yeah. And then with this assay, I mean, I really like your approach that you come from the procoagulant and the fibrinolytic pathway. And you also mentioned before that it could maybe help also profiling a patient or help the treatment. But you also mentioned in the beginning of your discussion that it's quite labor intensive. So how would you imagine that being implemented as a sort of diagnostic tool or helping the patients?
Yeah, I think in that matter, I think we can stick to this IMS strategy like immunomagnetic, which is like a very reliable and a very fast approach as like compared to conventional EV isolation method. Because in the clinical settings, I think it's very difficult to use this laborious ultracentrifugation, which takes several hours, or when we incorporate other methods like sec column, it's a very time consuming. So I think we can definitely use as like IMLs, but given that it needs further optimization, and we are actually like doing that as well back in Marseille and other.
Thank you.
Yeah, Akhil Konkoth, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return.
So I leave?
No.
we leave, Akhil. Well done. Oh, okay. Now something happened.
Okay, I see everyone is back. Then Akhil Konkoth, the committee here present online has assessed the quality of your thesis and of your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Koene is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Thank you, dear Prorector. So before I officially hand over the thesis virtually, I would like you to make a promise. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes.
Thank you very much. By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Akhil Antony Konkoth, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor, affixed with the official seal of the university, as shown now on screen by the beadle. I'm going to make a screenshot. Thank you congratulations,
Yeah.
dear candidate, Dr. Konkoth. Shall I proceed immediately with the laudatio? Yes. Dear Dr. Konkoth, dear Akhil, although it was already allowed to call you doctor, since you've successfully obtained your PhD under the expert supervision of Professor Romaric Lacroix and Professor Françoise Dina-Georges in Marseille in March, I'm still delighted to hereby also congratulate you and your family and friends with this achievement also here at Maastricht University. I can still remember the application procedure we started for the TICADIO Consortium back in 2019. We had to screen a lot of candidates, all by video conference, as now. The candidates were truly international, coming from the Far East and the Far West, and there were those who made a positive impression and others. You struck us as a friendly and committed person. You were somewhere in the UK, I believe it was in Scotland. You were already doing your PhD, and although the reason was unclear to me, you were looking for a new challenge. We were positive about you because you had experience with techniques on extracellular vesicles, next to the good impression that you made as a person. But we nevertheless agreed on a second round of screening. For some reason, I never witnessed a second round because I received an email in the meantime from Françoise that read something like, Rory, we have had contact with Akhil again and we decided to hire him. He's great. So with this, your journey in the world of extracellular vesicles continued in France. We are talking beginning of 2020, and all of us know what this meant. A dark cloud of virus-related misery amplified by the in part hysterical reactions of media and politicians passed over the world and made no halt at our universities. It also delayed our project, but my impression was that at least, judging from the outside, you kept your cool and just continued doing your thing. During this time, we had regular contact over Zoom, and we were preparing your visit at biochemistry in Maastricht. I believe this visit was quite delayed, but it was not a bad thing, since it gave you time to prepare your data in a way that we knew quite well what needed to be done in Maastricht. And although we had prepared your visit to be in early 2022 well, your arrival turned out to be unexpected after all. This might have had to do with a very pleasant event in your life, and this was your marriage at your home in India in 2021. It might be because of the subsequent period period of honeymoon bliss that the communication between us broke down for some time, only for me to find out that you were practically standing before our front door when you wrote me the first email afterwards. My plans were that you would be instructed by Alex here in this illustrious round of the corona for the work to be done. But Alex was outbound herself, and so it was up to me to guide you into the art of immune and inflammation assays. Luckily, you were a quick learner, and you had a talent for mastering also the more difficult assays. I think I can speak on behalf of my colleagues that we all enjoyed your laid-back and sympathetic nature, which, if I can correctly recall, made your supervisor Tillman call you the dude, which, if you know him, is a fine compliment. Despite your dudeness, you were quite on top of things, and you managed to finish more of our planned planned experiments during your 6-month stay. From this time, I noticed that the project picked up quite well, and we currently have your main manuscript on the role of extracellular vesicles from different cell types in the balance of coagulation and fibrinolysis during sepsis, currently under revision in the well-esteemed Journal of Extracellular Vesicles, with good hopes for a successful closure. And perhaps we can continue to work again to bring also the inflammatory aspect of these vesicles to a publication. I look forward to this. You are continuing your career in Birmingham at the Institute of Cardiovascular Sciences in the lab of Professor Asif Iqbal. I'm convinced that the many things you have learned under the supervision of Romaric and Françoise, and from me, will help you to further shape your successful career and to be an at least equally valuable colleague in the Birmingham lab as you were in our institutes. Having said this, I wish to take the occasion to thank the members of the reading committee, the Corona members, and the Pro-Rector for taking part in this online defense and its preparation, and to congratulate you and your family again with this achievement. All the best to you, dear Akhil.
Thank you. That was nice. Thank you.
Dear Dr. Konkoth, also on behalf of Maastricht University, I would like to congratulate you with the degree you have acquired. It's an important day, and it's a pity that you are not able to attend it here live, because that would be much nicer, but it's good that we can do it this way. I also like to congratulate your family, your wife, friends and colleagues with this important step you've taken in your academic career. I also like to congratulate your supervisors with this important day, and I'd like also to thank, on behalf of Maastricht University, the members of the degree committee for the time they invested in preparing and being present right now. Thank you very much. Well, I can say a little bit from the deliberations we had about the way you defended your thesis. I think everyone was happy with the way you defended your thesis and appreciate your enthusiasm and your knowledge on the topic. And the committee is sure that you will go ahead in your academic career and, well, go for the next milestone, maybe. I also hope that you look back on this defense ceremony with satisfaction, and I know that sometimes takes some time, because you always think, oh, I should have said this or I should have said that, but that doesn't matter. It's now time to enjoy, and I hope you have time today to also celebrate this special day, and I wish you all the best in your future career. And with this, I close this academic ceremony.
Thank you. Thanks, all.
Okay.
Maybe if someone wants to say something
Congratulations.
to the candidate.